Okay everyone, here we are for the third part of the lecture video for Chapter 5 on large biological molecules. If you remember, we ended on this slide here. In the second part, we were talking about the different functions of proteins. There are about 6 to 8 different categories of protein function. We'll go back through those. Those were covered in the previous video. What we are going to do next, though, is begin to talk about protein structure, so how it is that proteins are built. Remember, we've talked about this already for carbohydrates. We then moved on to lipids, which are fats and oils. Carbohydrates, we said, were built from the monomer-polymer plan, monomers being small building blocks that we link together, usually in a chain or in a branched pattern, and that produces a polymer, okay? We got away from that with the fats. Remember, they are the one category of biological molecules not built on that monomer-polymer plan. But now we're going to get back to monomer-polymer with our proteins. Okay, so proteins are all built from monomers called amino acids. And most people have heard of amino acids, whether they really know what they are or not. Amino acids are small building block molecules specifically used to build proteins. You only find them in proteins. They're not the building blocks of DNA or sugars or anything else, all right? And there are 20 of these. There's a set of 20 amino acids that all of our proteins are built from. Okay, so if amino acids are the monomers that are linked together to build proteins, okay, what do we call the polymers of amino acids linked together? What do we call the amino acid chain? Well, that's called a polypeptide. So a polypeptide is a chain of amino acids, right? And the reason why we call it peptide, in other words, polypeptide, many peptide, is because peptide is the kind of bond between the amino acids, as we'll see in a minute, okay? So polypeptides are unbranched polymers built from amino acids, this set of 20 amino acids in varying combinations. A protein, then, is a biologically functional molecule that consists of one or more polypeptides. A polypeptide, a chain of amino acids, in and of itself is not a protein. It has to, first of all, fold up into a very specific three-dimensional shape, and then it may actually join up with other polypeptides that have folded in similar ways, and only then do you get a functional protein. So it's important to make the distinction between a polypeptide and a protein, okay? All right, so let's take a look. I can get my slide to advance. There we go. All right. So here we have our list of amino acids. Now, these are all 20, okay? And what you'll notice in these is that every amino acid has the same basic structure. So I'm going to draw that down here in the right-hand column. What you have in the center is a central carbon called the alpha carbon. All right, write that up there. That's the alpha carbon. And then attached to that, you have a hydrogen. Then on one side, you have a carboxyl group, one of the 7 functional groups we've already talked about. On the other side, you have an amino group, NH2, also one of the 7 functional groups we talked about. Then, finally, the fourth bond of the carbon is made up by what we call an R group. So, the R group is what varies from one amino acid to the next. Everything I'm circling here with a pointer is common to every amino acid. They all have this structure. It's the R group that makes them all different from each other. So take a look, for example. You can see that here we have a grouping of amino acids right here, okay, about 9 of them, that are all nonpolar. So right now off the bat, you should know they're hydrophobic. That should tell you that they don't dissolve in water. They don't attract to water. All right, so let's take a look and see what's similar here. Well, everything highlighted in purple is what I've shown you right here, the alpha carbon, the carboxyl group, the amino group, and the hydrogen. Now, I wish they wouldn't do this, but they have. In your textbook, they've shown these in ionized form. So you have alpha carbon, hydrogen, and then you have a carboxyl here, but notice it's missing a hydrogen. That's because the hydrogen, which is usually there in the oxygen, has broken away and come over here and attached onto the NH2, making it NH3 plus, leaving this as O minus. Remember, oxygen being electronegative steals the electron of hydrogen when hydrogen breaks away. It went over here, hydrogen is now a proton, makes NH3 plus, leaving O minus there. Anyway, this is the ionized form. They're showing them all this way. It's exactly what I'm showing you down here. Just think about this down here and you'll be fine. All right, so this is all consistent with all 20 amino acids. As you see, everything in purple is the same. But here we have side chains, or R groups rather. In this case, it's just a hydrogen. Here it's a CH3 group, methyl group. Here you have this group, this one, this one. All of these R groups are different from one to the next, but in this case, as far as this group goes, all of these R groups are hydrophobic, nonpolar. That means that those amino acids are also nonpolar. They don't attract to water. That's critically important, as we'll see in a little bit. All right, also notice we have the 3-letter abbreviation, which is much more commonly used than the single-letter abbreviation. So glycine is GLY, alanine ALA, so on down the line. Here we have a group of polar amino acids. Their R groups are polar, which means they're hydrophilic, they attract to water. So these amino acids attract to water. Here we have amino acids that are unusually acidic. They donate protons extremely well to solution, driving the pH down. Here we have amino acids that are extremely basic. They take up protons, H plus, from solution and drive the pH up. All right, so this is really very important in terms of understanding overall protein structure. You can think of it this way: if you have a chain of, let's say, 100 amino acids, a polypeptide that's 100 amino acids long, and maybe the 25 on one end are roughly hydrophilic, the 25 on the other end are roughly hydrophilic, let's say that roughly the 50 or so in the middle are hydrophobic. You can make 100 million copies of that exact same sequence of amino acids, put them all in water at the same temperature, same pH, and they will fold up in precisely the same three-dimensional shape every single time. The reason for that is because the 50 or so in the center, in our example, that are hydrophobic, sort of collapse down onto themselves in the center, trying to get away from water. Think about oil and water not attracting to each other or separating. It's the same thing. The 25 amino acids on either end that are largely hydrophilic will wrap around that hydrophobic core to protect it from water. So it's these interactions with water, hydrophilic, hydrophobic, that will cause that polypeptide to fold up. But now if you change that polypeptide sequence, the amino acid sequence a little bit, swap out some hydrophobic amino acids for some hydrophilics and vice versa, that thing will fold up in a different way because now it's going to interact differently with water. And so ultimately it's the amino acid sequence and whether or not the amino acids are hydrophobic or hydrophilic that will cause that polypeptide to fold up in the way that it does. All right. So we said that amino acids are linked by covalent bonds called peptide bonds. Okay, peptide bonds. That's the bond between two amino acids that links them together. A polypeptide, therefore, is a polymer of amino acids. Polypeptide, many peptide bonds. And these polypeptides can range in length from maybe just a few to more than 1,000 or so in many cases. And again, each polypeptide for each amino acid will have a unique linear sequence of amino acids. So let's take a look at how a peptide bond forms. All right, recall that earlier in the chapter we talked about hydrolysis reactions and condensation reactions. Hydrolysis reactions separate monomers from each other by breaking bonds, hydrolysis, water breaking, and condensation reactions create bonds between monomers by producing a water molecule. Well, here we have a condensation reaction between one amino acid and two other amino acids already linked by a peptide bond. First of all, peptide bond shown in blue here is always between the carbon of one carboxyl group of one amino acid and the nitrogen of the amino group of the adjacent. It's always a carbon-nitrogen bond. That's a peptide bond. All right, here we have a new amino acid coming in. There's its amino group. Here we have the carboxyl group, COOH, of the next amino acid, and because they're functional groups, the reaction will always be between them. So the hydrogen here, the OH here, join up, separate away as water, and you get a bond between that nitrogen and that carbon. Right there. That's it. That's our peptide bond. And then you could have another amino acid come in with its amino group, joining up with that carboxyl group, and you have exactly the same thing occur there. So that's how these amino acids are linked together, by peptide bonds. Okay, so now we're going to get into talking about protein structure and function. And this is critically important for understanding how proteins work, how they do their jobs, because this is a really important part of biochemistry in terms of understanding structure and function. Structure and function really comes into play here. Proteins do precisely what they do, perform their functions, because of their three-dimensional shape. And their three-dimensional shape is a direct result of the amino acid sequence of the polypeptide that they're made of. So the specific activities of proteins result from their very intricate, at times, three-dimensional architecture, or the way that they're built. A functional protein consists of one or more, sometimes two or three, polypeptides that are precisely twisted, folded, and coiled into a unique shape. So we've got an example here. So this is a protein. I forget exactly which one it is, but it's the same protein shown in three different ways. Okay, so here we have a ribbon model on the left, and what you're seeing is the polypeptide. That's the chain of amino acids just shown as a ribbon. You can see how it has a very particular three-dimensional shape with a groove here, for example, sort of a depression on its surface. This is a space-filling model showing you the individual atoms. It gives you a better idea of the overall surface contours. You can see our groove right there. This is a wireframe model, just basically showing you the carbon framework of that. And again, you see we have sort of a groove or depression here. This groove or depression on this protein is a space for another molecule, a target molecule, to fit, like a key going into a lock. And this is generally how enzymes work. So what you're seeing here is actually a protein that's classified as an enzyme. It causes a reaction to occur in some target molecule that binds to it. We'll see more about that as we go forward here. So we've said that the sequence of amino acids determines a protein's three-dimensional structure, and that structure determines how that protein works, okay? So the reason why the shape of the protein determines how it works is because the function of the protein depends on its ability to recognize and bind to some other molecule. The key to your front door, or to any lock in any door, works because the key, the structure of the key, has a relationship with the structure inside the lock on the door. If you take a hammer and knock off a tooth from your house key, it isn't going to work anymore because you've altered its shape. And the same is true of proteins. If their structure rather is altered, they're not likely to perform their functions anymore because generally they fit together with another molecule in a very precise way. Change that shape, change that structure, molecules may not fit together anymore. All right, so we have basically 4 different levels of protein structure, which I'm going to show you part of here with this video in just a second, okay? The first one is called primary structure. Primary structure. This is nothing more than the sequence of amino acids that we've already talked about. If you have a polypeptide of 100 amino acids and you read the names of those amino acids from one end to the other, you are defining the primary structure, okay, by reading off the sequence of amino acids. That primary structure then will then fold up into a more complex way into what's called secondary structure, okay, and this is also partially due to its interactions with water. The secondary structure can then fold up into a third level of structure called tertiary, and we'll see these names here in just a moment, okay. Tertiary structure is determined by the interactions among the various R groups of the amino acids. Finally, you may have one or more polypeptides that have joined up or folded up into a tertiary structure that then come together as a quaternary or fourth level of structure. We're going to see details about all these as we go forward, so let me go ahead and play this video.
Each protein has a unique primary structure, a particular number and sequence of amino acids making up the polypeptide chain. 20 different amino acids are used to build proteins. Theoretically, the various amino acids could be linked in almost any sequence, forming an almost infinite variety of different proteins. This illustration shows some of the amino acids making up the primary structure of a protein. The structure of a single generalized amino acid is shown below. The main backbone of every amino acid is the same. This is what forms the backbone of the polypeptide chain. It is the R group, which projects out from the backbone, that makes each of the 20 kinds of amino acids unique. Different amino acids have different properties that affect the folding of a protein. Thus, primary structure ultimately determines the shape of a protein, which determines its function.
Okay, so a couple of things I hadn't mentioned here in terms of primary structure. Remember that what's common to every amino acid, okay? In other words, what I'm highlighting right here, this makes up the backbone, right? The backbone. So you see the nitrogen of an amino group here. You see the carbon of a carboxyl group there. Nitrogen, carbon. Remember, these are our peptide bonds, right? So the links then, the peptide bonds form the backbone, and then the R groups come off to the side in what's called a side chain. And because these R groups are different, they will interact with water, and that will cause this polypeptide rather to fold up in certain ways. Okay.
Each protein has a unique.
Sorry. Messed that up. Okay. So, sorry guys, the iPad's a little bit touchy on some of this stuff. Okay, so here we see a slide showing us the primary structure. So we have a polypeptide here, okay, and this one maybe has 127 or so amino acids in it, something like that. You can see how they're all linked together, like we said, by peptide bonds. There's one, there's another, and so on. And so this amino acid, glycine, has the amino group coming off of that end. That means that this end of the polypeptide is the amino end. Follow it down, protein, threonine, glycine, glutamic acid, and so on, all the way down the line. You get to this final amino acid, which has a carboxyl group coming off that end, so this end of the polypeptide is identified as the carboxyl end. All right, so that's typical length for a polypeptide. Put that in water and it will fold up in a very specific way because of the interactions of the R groups with water. All right, now, how does it fold up? Well, the first thing that happens is it folds up into what's called secondary structure, all right? So here we have secondary structure. There are two types, what's called an alpha helix that you see here, and a beta pleated sheet. Okay, so Greek letters, alpha, beta. Okay, so imagine that you have a polypeptide and it's big enough you're holding it in your hands, right? And you have, your hands are holding either end of it, and you take it and you twist either end in opposite directions. What's going to happen? Imagine what's going to happen. It's going to fold up into sort of a spiral structure, almost like a coil or a spring or a spiral staircase. That's exactly what an alpha helix is. And notice, let me zoom in a little bit, that in some areas you have hydrogen bonds between hydrogen atoms and oxygen and nitrogen in other parts of the molecule. These hydrogen bonds, while being weak, because there are so many of them, they can strongly reinforce that alpha helix structure, all right? Now, beta pleated sheet, what's going on there? So imagine you take either end of the polypeptide and you sort of fold them over onto each other parallel. So you sort of do this with it, right? You sort of fold the areas over parallel. Well, then you'll get hydrogen bonding between certain amino acids that are now adjacent to each other like this, and overall it will form a pleated structure, almost the way that paper would look that you fold it into a fan, then you unfold it and it has that sort of zigzag pleat to it. That's what happens here when these hydrogen bonds form between adjacent amino acids. It's called a beta pleated sheet. Those are our two secondary structures. Now, the R groups then begin to interact with water, and you get all of this beta pleated sheet, alpha helix, and other areas of the polypeptide that are still just primary structure. It all sort of folds up or mashes up into a ball. That gives you tertiary structure, tertiary level. These are often called globular proteins, okay? Globular proteins. All right, so the globular protein then takes on that characteristic sort of topography or surface contour features based on how the amino acids interact with water, hydrophobic, hydrophilic. You can see in some areas you have alpha helices, in other areas you have pleated sheets like here. Still in some areas, like right there and like right there, you still have just primary structure that has not folded up into a pleated sheet or an alpha helix. But the whole thing sort of takes on a globular overall shape. Proteins have to achieve this level of structure to become active, to start doing or performing some particular function. And many of them form a tertiary structure, start doing their jobs, they don't fold up any further. But in some cases, you'll have several tertiaries that join up together to make a quaternary structure. Here we have one tertiary and a second tertiary. So that's a polypeptide folded up into a tertiary. That's a polypeptide folded up into a tertiary. They attach to each other, making a quaternary structure. Many proteins do actually take on quaternary structure, and these are some of the bigger proteins, like hemoglobin, for example, that we'll see here in a little bit, okay? All right, go back for a second to secondary structure. The coils and folds, right, the alpha helices, the pleated sheets of secondary structure result from the hydrogen bonds between repeating components of the polypeptide backbone, like I've shown you here. Hydrogen bonding is really important for forming secondary structure. All right, and again, those are called beta pleated sheets and alpha helices, okay? So again, we see this a little bit in a more close-up view here. Hydrogen bonding is really important at this level of structure. Helps to lock these shapes in place, prevent them from unfolding. That's important because if a protein unfolds, as we're going to see a little bit later on, it loses its function. It can no longer perform its job, so it has to maintain its shape. And at secondary level, hydrogen bonding is really important for that. All right? Okay, let me play a little video here.
In most proteins, parts of the polypeptide chain are coiled or folded, forming twists and corrugations. This is secondary structure. The turns and folds of secondary structure contribute to the protein's overall shape. One kind of secondary structure is the alpha helix, where the chain twists. Another is the pleated sheet, where the chain folds back on itself, or where two regions of the chain lie parallel to one another. Secondary structure results from hydrogen bonding between atoms along the polypeptide backbone. Oxygen and nitrogen atoms along the backbone are highly electronegative, giving them partial negative charges and leaving nearby hydrogen atoms with partial positive charges. These negatively and positively charged atoms are attracted to one another at regular intervals along the chain, causing parts of the protein to twist or fold back upon itself.
Okay, so that's the two types then of secondary structures. Now we're going to move on to tertiary structure, again, what we refer to sort of generally as globular proteins, okay? So in tertiary structure, the overall shape of a polypeptide results again from those interactions between the R groups, between each other and with water, all right? So tertiary structure is not about interactions between the backbone components, right? That's secondary structure. Those interactions are hydrogen bonds that you see here on this slide. Tertiary structure is about interactions between various R groups and those R groups and water. So we have hydrogen bonds going on here in tertiary structure. We have ionic bonds, hydrophobic interactions with water, hydrophilic interactions with water. So there are a number of different types of interactions between the R groups themselves and between them and water that will then cause that tertiary structure to form. We also have a new kind of bond we haven't talked about, as we'll see, called a disulfide bridge, okay, and you'll see that right here. So first of all, this is just showing you basically the backbone of a polypeptide, right? That's all the amino acids linked together by their peptide bonds right there. The R groups are coming off of these various amino acids, and highlighted in yellow, they're showing you all these different interactions. So, yes, we do have some hydrogen bonds that are important for forming tertiary structure. Here's one example. between that R group and that R group of adjacent amino acids. Hydrophobic interactions. Methyl groups, as we said before, CH3, are hydrophobic. They have no charges on them. So remember this, like dissolves like, right? So if you have oil, two types of oil, olive oil, let's say, and corn oil, you mix them together, they'll mix because they're both hydrophobic. They attract to each other. So these R groups being hydrophobic and these being hydrophobic, they attract to each other. That's a hydrophobic interaction, okay? Don't worry about the van der Waals interactions, not important here. Here, let's go down here first. Here we have an ionic bond. So we have an oxygen that's negatively charged. It's an ion. We have an ammonium group, NH3, which has positive ions in it, hydrogen ions. They attract to each other. That's an opposite charge attraction, ionic bond. That can help to lock this loop together, in addition to what's called a disulfide bridge. A disulfide bridge is a very strong covalent bond between two sulfur atoms, as you see here. So there are two amino acids. If you go back to that table of the 20 and look at them, there are two that have sulfhydryl groups. So sulfhydryl is SH. If they happen to be adjacent to each other, like this one that has a sulfhydryl and this one, the two sulfur atoms of the sulfhydryl groups will covalently bond together in a very strong covalent bond called the disulfide bridge, and that can help to lock that loop in place. All right. So we have a number of different interactions then here that help to cause tertiary structure to be what it is, and then once it forms, to lock it in place.
Superimposed on primary and secondary structure is tertiary structure, irregular loops and folds that give the protein its overall three-dimensional shape. The irregular folding of tertiary structure results from interactions among the R groups of amino acids. Acidic and basic R groups ionize, and these positively and negatively charged groups may form ionic bonds. Polar forces also contribute to tertiary structure. Hydrophilic or polar R groups may hydrogen bond with one another or turn outward and hydrogen bond with the surrounding water. Hydrophobic, nonpolar R groups cluster on the inside of the protein away from water. Tertiary structure may be further stabilized by strong covalent bonds between sulfur atoms in certain R groups.
Okay, so that's tertiary structure, and what you're seeing here is a globular protein, a tertiary level protein, okay, and the three-dimensional shape of that will give it some particular function to play in our cells. Now, as I said, a lot of proteins stop there. They don't fold up any further, become more complex. They get their function at tertiary structure. But then we have a lot of proteins that go on, take it one step further, and become even more complex still. They fold up or become part of what's called a quaternary structure. Quaternary structure results when two or more polypeptide chains join together to form one large macromolecule. Collagen in our skin is a good example of a fibrous protein that consists of three different polypeptides that are sort of coiled up together almost like a rope. Hemoglobin is a globular protein that consists of four polypeptides, what are called two alpha chains and two beta chains. Let's take a look at that. Oops, sorry.
Superimposed on primary and secondary.
Wrong one, sorry.
Some proteins consist of two or more polypeptide chains. The fourth level of protein structure, quaternary structure, results from the combination of two or more polypeptide subunits. Quaternary structure is stabilized by the same sorts of attractions that stabilize tertiary structure. Hemoglobin, the red oxygen-carrying protein of blood, is an example of a protein with quaternary structure. It consists of two kinds of polypeptide chains. Two of each, a total of four chains, make up each hemoglobin molecule.
All right, so let's take a look at hemoglobin here. So this is basically a ribbon model of hemoglobin, and it's a large molecule. Every hemoglobin, which is a protein in our blood that carries oxygen, can bind four oxygen molecules. All right, so let's take a look. So we have one alpha subunit shown in dark blue here, a second one over here. Each of these is a tertiary protein. we have 2 beta subunits shown in lighter blue, one here and one there. Each of those also is a tertiary protein. So this is 4 tertiary or globular proteins that have joined together to make up a quaternary level protein that we call hemoglobin. Now, notice in the middle, we have what's called a heme group. It's a heme group. It's a grouping of atoms that contain an iron atom at their center. It's the iron that binds the oxygen. If you've ever lost a tooth or had a cut in your mouth, you've tasted blood, you know it has that metallic tinge to it. You're tasting the iron atoms in the hemoglobin that binds the oxygen. Okay, and because the hemoglobin has 4 iron atoms, one in each of its 4 tertiary proteins that make it up, as the quaternary protein, it carries a lot of oxygen, and so it's great for that. It's found in our red blood cells, as you see here. Okay, so now we've gone through the 4 levels of protein structure. Now we're going to talk about what it is that actually determines protein shape and therefore structure and therefore function. So we've talked about primary structure, we've talked about interactions with water, R groups, that kind of thing that causes it to fold up the way that it does. But what is it that maintains that structure? It's the environment inside of our cells, okay? Basically, it's pH, salt concentration, and temperature. There are a few other things, but those are the 3 primary things that affect protein structure. As long as salt concentration, pH, and temperature are where they should be, within normal limits, your proteins maintain their shape. But it is possible for pH or temperature or salt concentration to go outside of normal bounds for various reasons, and that can cause a protein to unfold. When a protein unfolds, we say it denatures, okay, or goes through denaturation. So denaturation is a process where a protein can unfold. You can have a quaternary level protein unfold all the way back to primary structure, just a loose polypeptide that has totally lost its shape and therefore becomes biologically inactive. It no longer performs its function. It's not even technically a protein anymore because it doesn't have the right shape. Okay, so here's basically what happens. A normal protein, this is a tertiary or globular protein, has this particular shape that gives it a certain function. Let's say pH goes much above 7.4, 7.5, or below 6.9, something like that, or temperature goes above 105 in humans, 106. That protein can denature. It can unfold and go all the way back to just a polypeptide, primary structure, a loose chain of amino acids. This is why a high temperature, above 105, can be so dangerous, because it can cause proteins, particularly in your brain, to unfold or denature, and brain chemistry can be dramatically affected, which can result in death. That's why a high fever is so dangerous. That's why our body goes through physiological processes that help to maintain constant temperature, constant pH, that sort of thing. That's much of what our physiology is about, is maintaining those constant internal environments that we need, environmental conditions that we need, because those are the conditions our proteins need to maintain their shape and not denature. Now, let's say that conditions have gone awry and therefore caused our protein to denature. If they go back to normal, that protein can renature. It can go right back to its normal shape again, and assuming the cell hasn't died or the organism hasn't died as a result, that protein can begin to carry out its function again. All right, so I've been going about 35 minutes or so. I think this is a good place to stop. The last video for this, the 4th one, is going to be fairly short, but it's just going to be covering DNA and RNA, nucleic acids. Okay, so we'll get to those in the 4th and final video.